All right, and good morning from my kitchen. So we're going to talk a little bit about the macromolecules. I know you watched about 100 con videos. I'll try not to do that again. But before you do your next free response questions, I just wanted to review a couple of things. So if we look at the first ones here, we have amino acids. So these are proteins. So proteins are made of amino acids. So with the amino acids, we have the N-terminus and the C-terminus. Remember, it's the N-terminus because there's an N. There's a C-terminus because that's a C, right? So the amino acids are connected N-terminus to C-terminus, right? So this reaction right here that joins them together, remember that's a condensation reaction, or we can say this is or dehydration synthesis. Okay, so the R, right? So the R right here, remember that's our functional group. And this gives it its shape or properties. So R groups, we have R groups over here. You don't have to memorize the R groups, but notice something about them. So we have nonpolar and polar. Polar and charged are pretty much the same, right? And nonpolar, so nonpolar and no charge are similar too. But if you look at it, most of the nonpolar ones here, right? We have an H there, CH3, all CH3s. There's an SH. So most of these right here, these nonpolar ones, do not have extra electrons. So if you look at the polar ones over here, look at what we have. We have an OH, we have an OH, an OH, we have an O, we have an O. So when it has O's and OH's, right, many times it becomes, oh, I'm sorry, this was a no charge, this was negative charge and positive charge. So polars usually have oxygens, right, attached at the end, and the non-polars usually have CH3s or H's. Okay, so make sure you know that because it'll be important to answer the next free response questions. All right. Remember protein shape. So this is protein shape. So primary structure. So this is the primary structure is just the polypeptide. Right, so the polypeptide. If I change an amino acid, I change the primary structure. Okay, so the secondary structure. Remember, that's an alpha helix or beta pleat, right? If I change an amino acid, but I don't change the alpha helix or the beta pleat, I don't change the secondary structure, right? Over here, oops, get back to where we were, shrunk it for some reason. All right, so here we go. So if we look at its tertiary structure, right? Its tertiary structure, it's its conformation. So its tertiary structure is its conformation, right? And remember, again, that was a shape. And that gives it its function, right? If I change an amino acid, I have to change its tertiary structure. because its shape will fold differently. Okay. In a quaternary structure, remember this is two or more. Two or more proteins are what's called a globular protein. Okay, so if it has two or more proteins and I change an amino acid, I will change its quaternary structure as well, because I change the folding of at least one of the amino acids. Okay, so important to remember. So if you do carbs, so if we do carbohydrates, remember carbohydrates are only made of CH and O. So our monomers or our monosaccharides, glucose, galactose, and fructose, they all have the same formula. Right? So we can further say that our structure is CH2O and N is the multiple. Okay, so this is a multiple of 6. Right? Remember the bond between them. It's another condensation reaction, right? It's a glycosidic bond. The protein was called a peptide bond. Our polysaccharides right, our polysaccharides, right? We have starch. So starch, this is plant. Right? Glycogen is animal. Essentially the same thing. These are a bunch of glucoses put together, and then cellulose is plant again. Right? So different types of polysaccharides. Right, so we have lipids. Right, so our lipids, remember we have a glycerol, and these are 3. So this is called a triglyceride. Okay, so remember this is formed again by condensation. So another condensation reaction or dehydration synthesis that joins it. All right, so you could say that this fatty acid right here is saturated. It's saturated because there are no double bonds. Right. So this fatty acid right here would be called monounsaturated because it has one double bond. If it has many double bonds, it would be polyunsaturated. So saturated just means holding as many hydrogens as possible. Okay. So this is a phospholipid. Right? Not a triglyceride, phospholipid, because it has a phosphate. Right? So this is our polar head and our non-polar fatty acids, right? So our hydrophilic and our hydrophobic. So really quick, nucleotides. We don't want that. Stick with green. So our nucleotides, it wants me to go multicolored here, right? So remember our nucleotides have a phosphate and a sugar. So this right here is the backbone. So that's our backbone. And this is our functional group, so nitrogenous base. Right? So nitrogenous bases. So we could either have DNA, oops. So we can either have DNA or RNA. Remember the way that they differ. Let me make this a little smaller so it fits. We have DNA and RNA. Oops. Right, and the way that they differ is based on the 2-carbon. Right, so DNA is deoxy at the 2-carbon, RNA is not. That's why DNA only forms single-stranded, or RNA only forms single-stranded, DNA will do double. RNA can fold much greater than DNA can because it has that extra point of oxygen there. And then oxygen can make bonds, remember. So again, in DNA we have thymine, in RNA we have uracil. Really similar. Missing a couple things, right? So this is different on DNA than it is on RNA. Okay. So we have our bases, right? A and G. These are our purines. And C and T are remember Chargaff said A is roughly equal to T, and C is roughly equal to G. Because of complementary pairing, right? So we see that here in the middle of the DNA molecule. All right, again, we have our backbone, right, which is our phosphates and our deoxyriboses, and our R groups are all in the center, right, and they're joined by hydrogen bonds, right? And the structure they make is an alpha helix. Okay, well, hope that gives you enough to get going today. Good luck, and I hope everybody's well. Bye.